everyone, and welcome to today's live broadcast, Preclinical Tcell Immunotherapy Strategies for Overcoming Immunosuppression in Solid Tumors, presented by Dr. Kristin Anderson from the University of Washington and Fred Hutchinson Cancer Research Center. I am Kaylee Bach of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit thermofisher.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing this presentation in the slide window. To enlarge the window, click on the arrows at the top right-hand corner of the slide window. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window. Or report your problem by clicking on the Answer a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Dr. Kristin Anderson. Dr. Anderson is a senior postdoctoral researcher at the University of Washington and the Fred Hutchinson Cancer Research Center in Seattle. Her research investigates strategies for treating solid tumors with adoptive T-cell therapy. Currently, she is developing molecular engineering strategies to improve tumor eradication using genetically engineered cells in ovarian cancer, with the ultimate goal of translating her findings into treatment protocols for patients. For a full bio, please see the Speaker Biography tab at the top of your screen. Dr. Anderson, you may now begin your presentation.
Hi, everyone. Thank you again so much for being here today. I'm really excited to talk to you about some of our most recent work, basically engineering T cells to try to kill solid tumors. So as the introduction stated, my name is Kristin, and I'm a postdoc in Phil Greenberg's lab at the Fred Hutchinson Cancer Research Center in Seattle, Washington. And I'm affiliated with the Immunology Department at the University of Washington. And so we actually get to do some really cool immunology and immunotherapy in this setting because we've got an immense amount of experience out here. So when I joined Phil's lab, one of the projects that he asked me to start working on was to take this idea that T cells have this ability to kill cancer and that we can engineer them to be even better at it and try to make it work in solid tumors, because his lab had a lot of success making this an effective anti-tumor therapy in leukemias. And we really wanted to push this to make it something that we could take to the clinic in solid tumors as well. So before I jump into the data, I just want to confirm for everyone that I have no financial disclosures to make, no relationships to disclose. And the goals of our session today, the things I'm going to be focusing on, have a lot to do with considering preclinical models and how what we're studying is related to what's going on in human disease. So I'm going to talk a little bit about the immunocompetent mouse model that we've been working in for ovarian cancer and talk about how we're trying to use both analysis of human tissue samples as well as these mouse samples in order to better understand what kinds of immunosuppressive features there are in human tumors and how the mouse recapitulates some of those features and how we can actually use that to our advantage. So the two examples I'm going to talk about today are strategies where we used whole transcriptome biosystems in order to understand what similarities actually do exist between the mouse model we chose and human disease, and then validating all of that using immunohistochemistry. In addition to that, I'm also going to talk a little bit about ways that once we know what suppressive obstacles are in front of us, we can try to engineer or use combination strategies in order to overcome some of those obstacles in the microenvironment. And then finally, I'll give you one example of something we've been working on really recently that we're very excited about. One way that we have found can actually overcome some of those hurdles, some of those death signals in both ovarian and pancreatic cancer that we're getting some really exciting data about. So I want to go back and talk a little bit about why we actually started working on ovarian cancer. When I joined Phil's lab, one of the big hurdles in T-cell therapy and immunotherapy in general was that there was a lot of evidence that things like checkpoint blockade and engineered T-cells were really effective against things like leukemia. And checkpoint blockade was starting to show a lot of real exciting promise in a lot of different kinds of solid tumors, especially things like melanoma. But there were still some tumors that, no matter how hard we tried, didn't really show a lot of change. And so ovarian cancer is one of those. And ovarian cancer is an incredibly devastating disease because this is one where the vast majority of patients who are diagnosed are actually diagnosed at a really, really late stage of disease because the symptoms of ovarian cancer are quite nondescript. We're talking about things like feeling bloated, or needing to urinate frequently, or feeling really full, even though you may not have had a big meal. Things like that could be associated with lots of different medical issues. And so an ovarian cancer is not necessarily an assumption that someone would make right away. And so oftentimes, if someone has these symptoms, they don't go into the doctor right away, or if they do, it's not necessarily the first test that's done. So unfortunately, a lot of these patients are diagnosed very late. And when it's diagnosed very late, there's a fairly poor prognosis for those patients. So as you can see from the data on the screen, the later the stage of diagnosis, the lower the 5-year anticipated survival rate or outcome for those particular patients. So what we thought we could do was to take advantage of some of the really advanced engineering strategies that were out there in the field, and to try to engineer T cells to target antigens that were already present in these tumors to see if we could take someone's immune system and make it stronger, to make it better against ovarian cancer. So adoptive T-cell therapy is actually already in use in the clinic in several different formats. So for example, TIL therapy is the kind of therapy where we isolate tumor-infiltrating lymphocytes from someone's tumor, expand them in the lab, and then reinfuse them into the patient. Or you may know quite a bit about CAR T-cell therapy, so T-cells that are engineered to have a chimeric antigen receptor, which fuses the scFv or antibody binding portion of an antibody with T-cell signaling molecules to help a T-cell more effectively target tumor cells. But in our strategy, we are actually, and what I'm talking about today, we actually are engineering T-cells to have high affinity T-cell receptors. And one of the major reasons we focused on this strategy was because there are a good number of tumor antigens that are not expressed on the tumor cell surface. And that can be problematic because chimeric antigen receptors are excellent tools for targeting tumor cells that have a protein abundantly expressed on the cell surface. But at the moment, they're not great for targeting tumor cells that express tumor antigens intracellularly. And some of the antigens that we've chosen to target are things like transcription factors, or in the case of the antigen I'm going to talk to you about today, they're proteins that are frequently shed from the tumor cell surface, but the tumor cell can still be detected as a tumor cell by a T cell with a T cell receptor, because that antigen is still made as protein and then processed and presented on peptide MHC. So a T cell with a high affinity T cell receptor still has the potential to see and eradicate those tumor cells, even if the antigen itself is shed from the cell surface as a protein. So the antigen I'm going to talk to you about today is one called mesothelin. And this is one that we're really excited about because an NCI working group actually characterized this particular antigen as one that would be a really great target for a lot of different kinds of tumors. So our group, Tom Schmidt and Ingen Stromness in the lab, actually engineered T cells to have high affinity T cell receptors that targeted 2 different epitopes of mesothelin. And so I'm showing you tetramer staining. This is basically flow cytometry, where we stain these cells with a tetramer that is essentially peptide MHC. And in this case, these T cell receptors recognize HLA-A2, and then either the mesothelin 20 or the mesothelin 530 epitope. And so we can make these cells, we can make lots of them that express this T cell receptor and can recognize that appropriate antigen. And if we co-culture those T cells with an ovarian cancer cell line, for example, in this case, I'm showing you co-culture with OVCAR3, which is one tumor cell line that's been identified as being like high-grade serous ovarian cancer, we see that these T cells can actually induce Cleve caspase 3 in the targets. And cleaved caspase-3 is a nice readout of induction of cell death because it's one of the last caspases in the apoptotic pathway. So generally speaking, once it's cleaved, the vast majority of those cells are on their way to dying. So this is really exciting because we think we've got something that we can move forward with into the clinic. But we know that solid tumors present a vast array of obstacles to T cells of any kind, even ones that have been engineered. And so our big question was, all right, we've made these cells, but is it even possible that they'll do anything because of all of those immunosuppressive obstacles they're up against? So we hypothesized that indeed, we made these high affinity T cells, and we think they can actually slow tumor growth and prolong survival, but we needed an immunocompetent system in order to test that out. And the other thing we wanted to look for was whether there was any off-tumor on-target toxicity, because mesothelin is technically a self-protein, and we wanted to make sure that we weren't seeing any immunotoxicities in healthy tissues due to the T cells killing healthy cells accidentally. So we turned to this model called the ID8 VEGF model. So the way that this was generated many years ago was the group scraped epithelial cells off the surface of a B6 mouse ovary and grew them in culture until they transformed into a cancer cell line. And so then, in order to make this line even more like high-grade serous ovarian cancer, another group transduced it to overexpress vascular endothelial growth factor, or VEGF. And so that's the line I'm going to talk to you about today. So what we do is we take this line and we implant it intraperitoneally, and we let those tumors grow for 6, 8, 10 weeks. And what ultimately happens is these cells form nodules or tumors all throughout the peritoneal cavity of the animal, and they vastly resemble high-grade serous ovarian cancer that's already in a metastatic stage. So as you can see from the images on the screen, when we screen these mice using an ultrasound, we can find these tumor nodules throughout the cavity, and they're circled and highlighted by arrows, as well as we can find ascites. So in these mice, they will eventually develop terminal hemorrhagic ascites, and hemorrhagic ascites is actually a key characteristic feature of a lot of patients with high-grade serous ovarian cancer. So on the surface, this line seems to recapitulate a lot of the major features of advanced stage metastatic ovarian cancer. Now, because we're going to target mesothelin, we want to make sure that the mouse model accurately and nicely expresses mesothelin in order to even see if we can do these experiments. So we performed immunohistochemistry, staining for mesothelin. So you can see a sample of a human high-grade serous tumor on the left. So the brown staining is mesothelin, and the mouse sample is on the right. And we went even further to double check and make sure that the level of expression in the tumors was much higher than healthy tissue. So we took tissues from either a healthy B6 mouse or a mesothelin knockout mouse, or our ID8 tumors after they had been growing in mice. And we did a Western blot for mesothelin. And as you can see from the blot on the bottom of the screen, the 3 different tumor samples that we took all had much higher expression of mesothelin in the tumor than either the lung, the mesothelial lining of the lung, or the lining of the ovary. So this was encouraging because this suggested to us again that this was actually a safe thing to target. So in order to engineer our cells, we follow a strategy that's similar to what is used to engineer human cells for treatment in the clinic. So in this case, we're going to use mouse T cells. And in our experiments, we use the P14 strain of transgenic mouse. So this is a mouse that has been engineered so that all of the CD8 T cells have a T cell receptor for an epitope of LCMV, this virus, this mouse virus. And all of these cells also are expressing a congenic marker, so that when we engineer cells and put them in an animal, or even if we just engineer cells and are looking at them in the dish, we can find them because we can stain for this protein that's on the surface of the T cells. So we isolate splenocytes from the mice, the donor mice. We activate the T cells in vitro. So we combine the cells with anti-CD3 and anti-CD28 antibodies, and we give IL-2 to the culture. That way that supports T cell growth. And then we actually will transduce 2 times on the next 2 days with retrovirus containing whatever constructs we want to put in this case, our T cell receptor. And then we usually get transduction efficiencies anywhere between 50 and 85% after those first few steps. But if we want to enrich and increase the number of T cells that we have expressing our construct, we can actually do one more step where we irradiate some donor splenocytes and pulse them with our peptide of interest, in this case mesothelin, and then we can co-culture our transduced cells with those peptide pulsed splenocytes and expand the number that we have that are appropriately transduced with the construct we care about. So then we can do in vitro experiments and really test what these cells are able to do. So the first question that I had was whether or not these engineered T cells could even kill this ID8 VEGF cell line. And fortunately, we were lucky and very excited to see that they could. So here I'm showing cleaved caspase 3 staining, like I mentioned before. So cleaved caspase 3 expression in these ID8 VEGF cells after they've either been co-cultured with mesothelin T cells, which you can see in red, or OT1 T cells. So these are P14 T cells that we engineered to express the OT1 T cell receptor, which is a T cell receptor specific for ovalbumin, a chicken protein. So these are irrelevant cells. They shouldn't cause death in our target cell line. And we were happy to see that they did not. So we can quantitate this and see that it's significantly greater in, cleaved caspase 3 is significantly greater in the target cells after we've co-cultured them with antigen-specific T cells. And we also went one step further and we utilized this technology called the Excelligence instrument. And this is an instrument where you plate your target cells and they adhere to the bottom of the well. And this plate actually has electrodes and sends an electrical signal along the bottom of the well. So anytime your target cells adhere, it impedes that electrical signal, and the computer can read that out. And then when you add your targets and your targets induce death, or excuse me, your effector cells, when they induce death of your targets no longer adhere, and so they no longer impede that electrical signal. And that can be read out as an indirect reading of target cell death. So we did that as well and found that we can actually get even cleaner data when we go down to lower E to T ratios. So we confirmed in multiple assays that yes, in fact, our T cells that we engineer can kill this particular tumor cell line. So moving forward, we wanted to do some in vivo experiments where we looked at whether or not the T cells that we engineered could get into tumors, could cause death in these tumors, and could ultimately prolong survival of these mice. So the timeline for these experiments is much longer than what many people are using when they do in vivo assays. So this model, because it takes so long to develop, and because our goal is ultimately to model advanced stage metastatic disease, we actually inject these ID8 cells into the animals and then wait at least 6 or 7 weeks before we even start screening to see if the tumors are there. And usually we don't start treatment until anywhere between 6 to 8 weeks after tumor cell injection. So we're waiting until we can see those nodules by ultrasound. And so our goal here is not to necessarily clear tumor. It's simply to make sure that we set the bar high because we want to really mimic as much as possible what kinds of patients we would be seeing when we first go into the clinic in a clinical trial. So as soon as we found mice that have measurable or detectable disease, we transfer T cells intravenously, and then we give those T cells IL-2. So we give IL-2 injections subcutaneously for about 10 days in order to support T cell survival and proliferation. So this first experiment I'm going to show you, we just did one injection of T cells, and we took the mice down at various time points to simply ask the question, are these cells even able to get into these tumors? Because we know that a lot of solid tumors have physical barriers that prevent T cell infiltration. So in this case, we were excited to see that, yes, in fact, we do see T cell infiltration. So this is immunohistochemical staining for CD3. And as you can see in the very first panel, if we don't transfer any T cells, these tumors have very, very, very few T cells in them whatsoever. After we transfer T cells, we see an influx of T cells in the tumor. And if we pull these out and we actually do staining by flow cytometry, anywhere from 50 to 90% of these cells are our donor T cells. So most of them are CD8s. We don't seem to see an influx in Tregs. So most of them are CD8s and most of them are, in fact, our donor cells. But we notice as we let the mice go and we watch these T cells over time that they don't seem to be persisting. And this is not necessarily a big surprise. We are making our T cells into effector T cells. And the literature has very elegantly shown that T cells expand in the effector phase and then contract after antigen is cleared or after as they approach memory. So as they come out of that effector phase, we would expect that the cell numbers would go down. But unfortunately, we were seeing numbers very similar to what they looked like with no T cell transfer at all. So this was concerning, and I will actually come back to this at the end of the talk to talk about an obstacle that we wanted to overcome in these tumors. So our other question was, well, the T cells that are still present at these later time points, what do they look like? When we did phenotyping and functional analysis to see what these T cells looked like before we put them into the mice, they all have an activated effector phenotype, much like you'd expect. They have expression of CD69 and 4-1BB because they've recently seen antigen in the dish in vitro. But they also have slight upregulation of a lot of the molecules that we commonly consider to be inhibitory receptors, PD1, LAG3, TIM3, TIGIT. And so we put these cells in, we take them out, and see how the phenotype has changed. So when we pull these out at an early time point, we see that they still look activated. And when we pull them out at a late time point, this was the time point we were most interested in, we see that all of these markers are still highly expressed on the T cells, the donor T cells that we pull out of the tumor, relative to the donor cells we pull out of the spleen. And that was important to us because that suggested that these cells, it's consistent with them having a signature of being dysfunctional or immunosuppressed. So we wanted to see if functionally the cells could actually make anti-tumor cytokines. And so when we make these cells in vitro, screen before we put them in, they're very capable of making interferon gamma and TNF alpha. But when we pull them out of tumors at day 21, it's very clear that they have vastly lost this ability to make cytokines. So consistent with the phenotype, it seems like these T cells are definitely becoming dysfunctional. And we know from vast amounts of really awesome literature that there are many, many, many ways that the tumor microenvironment can cause T cells to become dysfunctional. And there's so many, I don't have time to get into all of them today. There are some really awesome reviews out there that you can read if you'd like to learn more about driving T cell dysfunction in the microenvironment. But we knew that all of these were possibilities. Any one of these or any combination of these could be the reason that our T cells are not retaining function and not persisting in the tumor microenvironment. So we wanted to ask which features of this mouse model we were using were similar to the features that were present in human ovarian cancer. That way we could better understand which obstacles we could actually study in the mouse disease. So we wanted to understand how much these tumors in the ID8 VEGF mouse model actually recapitulated features of human disease. And to do this, we used Thermo Fisher's Applied Biosystems Deep Transcriptome Profiling in order to truly understand what genes are being overexpressed and what pathways are the same or different between our preclinical model and real patient disease. So to do this, we got mouse samples from our tumor-bearing mice at late stages of disease, and we were able to get from a local repository some samples of human disease from patients, and these samples that we obtained were matched samples. So we have samples from 5 patients. We have their primary tumor. We also have a sample of metastatic disease from the same person, and we also have a matched normal tissue sample or adjacent normal from the same person. That way we can really start to ask questions about, does the mouse model look more like primary disease? Does it look more like metastatic disease? And having samples from the same matched patient actually makes the analysis nice and clean. So we got samples that were fresh frozen at surgery, so either at mouse euthanasia and necropsy, or in the clinic when the patient was undergoing debulking surgery. We did RNA extraction on all of these samples ourselves and did quality testing to see what the quality level was. And in the vast majority of cases, we had really good quality RNA, but there were a few samples that came back with slightly lower RIN values. And so when we sent our samples to the Thermo Fisher team, they actually were able to do quality control and checks on their end and told us that even though the RIN values and the quality was a little lower, that they thought the assay would still potentially work. And we were fortunate that it did. So we sent our samples to the services labs, and they performed the Applied Biosystems microarray for us. And what was really nice about this, we had them do the Clarium-D Pico assay. And we chose the PICO assay because we knew that some of the RNA samples we had from our particular tissues were quite limited in quantity. And so we wanted to do something that was incredibly good at low levels of input. And then ClariumD is one of the more recent microarray designs. And what's really nice about this one is that the probes actually they have great coverage of all the exons that you would care about, but they also cover splicing sites. So you can get a lot of information about different splice isoforms and expression of non-coding RNAs as well. So then for software analysis, we utilized both the Transcriptome Analysis Console, the TAC software that comes for free with the assay. And we used that to look a little bit at what our tumors actually looked like relative to normal samples. And we also used it to look at just gene expression overall, what genes were up or down in these tumors relative to normal tissue. And then we also used R not only to validate those findings, but also to look at how similar and how different the mouse and human samples were to one another in tumors. So first I wanted to show you a sample of what the TAC software actually looks like. So this is just a screenshot of one of my analyses. This is looking at the different human samples that we had run. And this is simply showing gene expression of a handful of genes from this particular array. And all of the different ways that you can analyze this data. So the QC view and summary view are really great because they give you sort of an overview of the quality of your samples and all of the different data that you got back. The gene view lets you dive much deeper into all the p-values, the FDR p-values, and figure out which genes are actually expressed or downregulated in each individual sample. And then one of the other really cool things that this program or this analysis lets you look at is the alternative splicing. So I'm not going to talk about that today because we didn't use it for this particular question, but it's one more really cool, powerful use of this particular assay. So in addition, you can take this gene expression information and you can plot it in heat maps and compare your samples and truly see where the patterns are. And you can export that and use it for figures if you need to. And again, one of the great things about the software that we really appreciated was that you could measure so many different things, even things that you might not have thought to ask about. So we were able to look at gene expression changes, which was our major question, but we were also, if we wanted, able to evaluate and dig down into splice variants and non-coding RNAs. And that coverage, especially of those low abundant transcripts, was really helpful for our particular analysis. So one of the main concerns I had and wanted to sort of evaluate before we went too far was whether or not the human samples that we had gotten from the repository looked like high-grade serous ovarian cancer. So high-grade serous is just one of many subtypes of ovarian cancer, and that's the subtype that this particular ID8 model is supposed to recapitulate. So high-grade serous ovarian cancer, in many patients, it's now becoming clear, actually doesn't derive from an ovary cell. It probably derives from a fallopian tube cell. And so one of the things we wanted to do was principal component analysis to see if our tumors looked more like fallopian tube or ovary. Now, in this example, we only had one normal ovary tissue to look at, but you can see from the principal component analysis that that green normal ovary sample looks much different from the red primary ovarian samples and primary ovarian tumor samples, as well as different from the metastatic tumor samples, and that the red and purple look much similar to the blue normal fallopian tube. So that was comforting because it suggests that what we are actually seeing is high-grade serous ovarian cancer. So even though the mouse model is derived from an ovary cell and the human disease is supposedly in many cases derived from fallopian tube, we wanted to know, is it still able to recapitulate the similar pathways, gene expression pathways in the microenvironment? So when we looked at our analysis and aligned all of these pathways side by side, we used the KEGG pathways, the KEGG database, in order to evaluate pathways that seem to be relevant or could be relevant for the efficacy of immunotherapy. So here I'm showing 98 of the pathways that we chose to analyze that came back as not statistically different, either between the mouse tumors and primary disease or between the mouse tumors and the metastatic human disease. So this was really exciting because most people are very skeptical about what transplantable models are actually simulating. And so the fact that these came back looking incredibly similar to human disease is really encouraging. It suggests that we might actually be able to use this to model quite a few of the obstacles that the human disease presents to immunotherapy. Now, that said, there definitely were some pathways that were not the same. And most of these came out as metabolic pathways, which isn't completely surprising. We know that this is a tumor cell line that is grown and maintained in vitro. So tumor cells, again, adapt very readily to their environment. And so it's not surprising that these cells have overexpression of lots of metabolic pathways. It just means that when we truly are evaluating our data from these mice, we'll have to keep that in mind, that the metabolic environment in these tumors is much harsher than what we will probably be up against in patient tumors. So going forward, we found all these pathways that were very, very similar, and several pathways that were different. But ultimately, because most of the pathways that were similar were the ones that were incredibly relevant for things like suppressive cells or antigen processing and presentation, we decided this seemed like a good model to keep moving forward with in terms of evaluating whether or not adoptive T-cell therapy with engineered cells could prolong survival in these advanced stage mice. So we did go back and try to validate some of these immunosuppressive features using immunohistochemistry. So here I'm showing, for example, staining for myeloid cells, which are a very well-known immunosuppressive cell in tumor microenvironments. So this is data both from human high-grade serous samples as well as the mouse ID8 VEGF tumor. These are tumors, again, that were grown in these mice for at least 8 to 10 weeks before we took these samples. And we've seen similar data for regulatory T cells as well. These suppressive cells are definitely present in both systems. And we've also seen that both human and mouse disease overexpress or at least have abundant expression of the ligands that are proposed for some of those inhibitory receptors that I showed you before are present on our T cells when we pull them out after they've been in tumors for about 21 plus days. So, for example, galectin-9 has been proposed as one of the ligands for TIM3, and galectin-3 is one of the ligands proposed binding to LAG3, which both send negative signals to T cells. So armed with this, and armed with some preliminary data that suggested that a single dose of T cells did not prolong survival at all, we decided to go at this really and hit it as hard as we could. So we made our T cells, and this time, instead of just injecting one dose, we decided, well, these T cells are becoming dysfunctional fairly quickly, so let's give fresh T cells often. Let's give fresh T cells every 14 days so that we are constantly hitting the tumors with effector T cells that have the potential to kill them. And in addition, instead of just giving T cells, we're going to give T cells and boost those T cells in vivo. So this is actually a protocol that was pioneered in a cancer cell paper by one of my colleagues, Ingrid Straumes, in 2015. And so we essentially recapitulated or utilized that treatment technique in these mice to see if it would prolong survival. So again, we wait until there's detectable disease at a late stage, and then we give a lymphodepletion regimen to simulate what often happens in the clinic. This has also been shown in the literature to promote cell persistence, donor cell persistence. So we tried treating with Cytoxan to lymphodeplete prior to T cell treatment. And then when we give T cells this time, we give T cells with peptide pulsed irradiated splenocytes. That way we're giving those T cells a quick antigen boost in vivo to try to expand them and increase those numbers in the animal. So we only give the lymphodepletion regimen once before the first treatment. After that, we give fresh T cells and this vaccine boost every 14 days. And we followed the mice to look at how long they would survive. And in this case, I will note that we are giving IL-2 every time we give T cells for 10 days, just to make sure that we're supporting the survival and proliferation of those cells in vivo. And we were really excited to see that this does significantly prolong survival of the mice. When we treat with, as you can see by the red bar on the survival plot on the screen, and what we were really excited to see was that if we actually take these tumors out of the mice after 3 T-cell treatment doses and look for cleaved caspase-3 in these tumors, we see that it is expressed at much higher levels in the mice that are treated with tumor-specific T-cells than in either untreated mice or mice that are treated with an irrelevant T cell. So this was really exciting. It suggests that our T cells are truly making a difference when we put them in the animals that are bearing tumors. And now our real question is, can we make it better? So like I mentioned earlier in the talk, one of the things we really wanted to do was see if we could identify obstacles to T cell efficacy and then figure out a way around them. So the example I'm going to show you today has to do with altered T cell trafficking and survival. So we mentioned earlier that the T cells we're putting in weren't persisting. And this could be for a number of reasons. But one of the reasons we found in the literature that we wanted to explore further is actually published by George Kukos's group several years ago. And they showed that in high-grade serous ovarian cancer, there's actually this increased expression of Fas ligand, which sends a death signal to T cells. And it's expressed at high levels on the tumor vasculature. And they showed that the mouse ID8 VEGF model also overexpresses Fas ligand on the tumor vasculature. So this gave us an idea. We thought, what if we can somehow take that Fas ligand signal, and instead of manipulating the tumor itself, what if we can engineer the T cells to see that death signal, but to see the signal in a different way? So I teamed up with a colleague of mine in the lab, Dr. Shannon Oda, who's an excellent molecular biologist. And she engineered this panel of what we're calling immunomodulatory fusion proteins. So there's an ectodomain that is the Fas receptor. So this is where the molecule actually will see or recognize that Fas ligand signal. But instead of having the intracellular domains that would send a death signal, she fused it with molecules that will send a positive signal, something that will either support proliferation or survival, to try to act both as a dominant negative, so that the T cells are ignoring or not receiving that death signal, but also to override it and send even more of a positive supportive signal to the T cells. So essentially trying to use the tumor evasion mechanism against itself. So we did several different versions of this construct. As you can see on the screen, I'm just showing you a couple of examples. We actually tested a really huge array of these molecules, and we narrowed it down to some that we thought had the most potential. And so here I'm showing you CD28 and 4-1BB, which have actually been talked about in the literature before as switch signals or ways that you can send a positive signal to T cells. And we did a lot of in vitro experiments to try to identify which of these had the greatest potential in vivo. And for the sake of time, I'm not going to show you all of those experiments, but I'll tell you that it looked like, based on the preliminary experiments that we did, the Fas 4-1BB constructs looked much stronger. So in terms of cytokine production and proliferation and survival and transcription factor expression, all of those were consistently better in the Fas 4-1BB cells. So we took that into our mice and tried to add this to the survival curve to see how cells that express both the mesothelin TCR that we had engineered, as well as this immunomodulatory fusion protein, could function in vivo. And we were really excited to see that when we include one of these Fas 4-1BB constructs, we actually see even greater prolonging of survival than if we simply treat with mesothelin T cells alone. Now, one construct worked much better than the other. One didn't seem to do anything in vivo, suggesting that that molecular biology that you do to make these constructs really matters quite a bit. But again, we were able to overcome an obstacle that we had identified in the tumor microenvironment and essentially figure out a way around it and make our therapy better because we made an informed set of experiments mimicking something that is present in the mouse model that we know is relevant for human disease. So in conclusion, we've covered a lot of ground today, but I wanted you to walk away knowing that this ID8 VEGF model actually does recapitulate many of the key features or hallmarks of human high-grade serous ovarian cancer, including the expression of a lot of these immunosuppressive features that are problematic and pose a lot of limitations or hurdles for immunotherapy of many kinds. And then also that if you identify an immunocompetent preclinical model that does characterize or recapitulate features of human disease, you have this ability to then design therapeutic strategies that will get around some of those. And those potentially have increased relevance for translation into the clinic. So with that, I want to thank all of the people that contributed to this work. I have a great team here at the Fred Hutch of technicians and medical fellows and undergrads that all work incredibly hard to get these experiments done. I also want to thank my mentor, Phil Greenberg, without whom none of this project just wouldn't exist. Shannon Oda, Dr. Shannon Oda, for all of her help making these fusion proteins, because without her, this last avenue of the project wouldn't be what it is. And then again, to all of the people who gave us technologies and samples so that we could do the really great work that we're doing. And obviously to the funding sources, because without them, this work wouldn't exist either. So with that, I am happy to take any questions that you have on what we've done so far.
Thank you, Dr. Anderson, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is the target antigen a self-protein, and did you see the toxicity?
That's a great question, and one that we do get all the time, so thank you very much for bringing that up. So mesothelin is actually a self-protein, and that's actually one of the reasons that we chose to target it, is because an NCI working group selected this as a really good cancer target antigen, simply because while it is expressed in some healthy tissues, it's actually expressed at a very, very low level. And in many, many tumors, it's expressed sometimes 10 or hundreds of times greater. So that actually makes it a good antigen because the chances of on-target off-tumor toxicity are fairly low. So we've been keeping an eye on this because, as you might expect, we are suspicious that targeting a self-protein could cause toxicities or pathologies in tissues that we don't want it to. And fortunately, in all of the pathology that we have done, all of the basically H&E slides that we have sent, all of the pathologists we've talked to in multiple different preclinical models, we haven't seen any toxicities. So while we have seen engineered T cells in tissues that have mesothelin expressed at normal, healthy levels, we haven't seen any pathologies or any mice that have succumbed to pathological issues due to T cell treatment.
Wonderful, thank you. Our next question says, did Clariome provide you with enough transcriptome information for your project?
Yes, actually, the Clariome D assay that we had performed provided us with above and beyond what we actually needed for our basic question. So when we got our data back, not only got data on gene expression, but we were also able to get information on isoforms and splice variants and things that we are going to be mining for years to come because we got so much data out of this one assay.
Our next question says, how was your experience with tax software for data analysis? How easy was it to learn and use?
That's a great question. So I am not a bioinformatics expert. My training was in immunology as a graduate student, and then in cancer biology and molecular biology as a postdoc. And so I actually leaned on Thermo Fisher and the tax software team to learn it the first time around. They were kind enough to send out a rep to teach me for free how to use the software, to talk me through all of the logistics, and left me with lots of notes and information that I could basically teach myself. So when I finally sat down and worked through the software on my own, it was really intuitive and actually very easy to use. So I only enlisted a bioinformatics expert to make sure that I had done it right, and was happy to find out that I had. So.
Thank you. What kind of combination therapies are most compatible with engineered T cell therapy?
That's a great question, and one that we're still actively investigating. As I showed in the presentation, we think that further engineering additional components of cellular, additional components of T cells is very powerful. But we think that this could actually be combined with many different types of techniques. For example, combining this with checkpoint blockade, something that's already in the clinic, has great potential for making these cells more functional in tumors much longer than they currently are.
All right, we have time for one more question, and this one says, why did you use a service lab to process your samples? How does sample submission work?
So sample submission was very, very simple. We contacted the representative and simply said, we have X number of samples, we would like to do this assay. And they sent us a plate, everything we needed to ship them our samples. And they did all the quality testing. And that was actually one of the major reasons that we wanted to enlist the team to help us with this. No one in my group is an expert at deep transcriptome profiling. And we wanted to make sure the samples were so valuable and because we had such a small amount of input that not only were we not going to make mistakes and lose valuable sample, but also that we had a thorough QC done to make sure that the data that we got back was really, really good quality data. So we had a great experience. The team was really helpful, easy to communicate with, and they were really fast. The turnaround time was really impressive.
Thank you again, Dr. Anderson. Do you have any final comments for our audience?
Just thank you so much for your attendance today, and please feel free to reach out if you have any further questions.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we do not have time for today or those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand through October 2018. LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.